We've got 3 chapters in microbiology to cover today, so I'm going to try to get through all of them. And of course, if you have any questions, you can drop it into the chat box, and I'll periodically check that. There we go. All right, so this is chapter 20, Introduction to Microbiology. And before modern science, there was a lot of magical thinking about the way the world worked. And so there was this theory called spontaneous generation. In the 17th century, scientists believed that living matter just spontaneously appeared from non living matter. And this theory came from even the earliest philosophers as far back as Aristotle. So there was actually common beliefs that fleas appeared out of dust, that maggots just appeared out of rotting flesh. And so this man named Francesco Redi came along, and he wanted to disprove spontaneous generation. So what he did was is he took a jar or two. He took 2 jars, and in one jar he put some meat in it. Let's say I can't remember what type of meat. Let's just say it was a cut of steak into a jar, and he placed another steak in another jar. And for the control group, we'll say, he tightly secured a lid and closed it so it was sealed. And in the other one, he left it open. Oh, sorry, there was 3. So one jar. Let me go back. Sorry, I haven't had coffee. I actually just got up. So Francesco Redi wanted to prove that spontaneous generation wasn't real. So there were 3 jars. One, he left the meat open with no lid. The other one tightly sealed. And then the third jar, he basically put like a cheesecloth over it, just some cloth netting. And what happened is the jar that was open was covered in flies and maggots, of course, because you left meat out rotting. The sealed jar, there was no flies or maggots, and the jar covered with cloth netting had maggots on top of it. So the flies weren't actually able to get down in the jar, but they still laid eggs. So Reddy's hypothesis was that if there's no flies, there will not be maggots. So the results actually supported the hypothesis that spontaneous generation doesn't exist, but scientists were still not convinced. So then a man named John Needham came along in 1745. And his hypothesis was spontaneous generation can occur in the right conditions. So he took he took a flask and filled it with broth, like a soup broth, and he left it open for a period of time. And after a period of time, you could look in the broth, and there were like microorganisms floating around. Um, so he thought that this proved spontaneous generation because the microorganisms appeared. So he called them animalcules, like little animals that appeared. And then we have Lazzaro Spallanzani, who believed that Needham was on the right track, But he said that Needham went wrong when he exposed the broth to air. So he took two different flasks filled with broth. One of them he left open, which ended up having microorganisms in it, and the other flask was sealed with a cork, and of course, things did not appear in it. So his hypothesis was that air is responsible for spontaneous generation. Louis Pasteur set out to disprove the theory of spontaneous generation in a science contest in 1859. Um, so he used an S-shaped flask, and what this did was it would let in air, but okay, let's look. So with the S-shaped flask, you apply heat to the broth, you let it sit, and because of the shape of the flask, the handle there, or not the handle, but there was no bacteria present. And then he took another one and he applied heat and he removed the neck, and bacteria was able to get in and it was present. And then he applied heat, tilted it sideways, and let it sit, and bacteria was present. So he tested the counter hypothesis developed by Rudolf Virchow in 1858 that living things can only come from living things. And this was a theory called biogenesis that had come from Rudolf Virchow. So Louis Pasteur was setting out to prove it right. And the conclusion was that microorganisms are everywhere, even the air. So science had finally realized that there were microorganisms. They cause disease. They arise from living things. They don't spontaneously appear. So another thing you need to know. These are some names that you need to know. So I'm just briefly going over them. Um, if you haven't got your book yet, make sure you get it next week because you want to be able to read along so you don't get behind. So, the first vaccination or the first vaccine that was made, well, there were other ones, but he got the first credit for it. So, Edward Jenner is who we're going to say um, made the smallpox vaccine. And what he did was, is he, wherever he was studying, he noticed that when there was a smallpox epidemic going around, the milkmaids never got sick. Um, and so what he did was, is he went to the milkmaids, um, and what happened is they would develop cowpox. And if you develop cowpox, you're immune to smallpox. So he basically like scraped off their cowpox blisters and got the germs out of that. And then he would inoculate healthy people, meaning they would ingest it or come into contact with it. And that was the first vaccine. Um, and so vaccine, the word vaccine comes from vaca, which means cow. And this is a theory of disease called germ theory. And this is what we still follow to this day. It's that microorganisms known as pathogens or germs cause disease. So these are a few names that you will need to know. Robert Hooke coined the term cells when he observed cork under a compound microscope. In the 1600s, he said that there were like little cells in it. Anton van Leeuwenhoek reported animalcules in rainwater, lake water, the surface of human teeth, horsefly guts, and human diarrhea. He basically just collected whatever he could um, to show these animalcules, which we have come to know as microorganisms, describe them. So you'll see whenever you take, we still have mats. Um, I think there's a mats question. Some places are going to describe Anton van Leeuwenhoek as the father of microbiology because of that. And a couple of more significant names, Agostino Bassi proved that a type of silkworm disease was caused by a fungus. Anton van Leeuwenhoek was also the first to see red blood cells under the microscope. And I'm going to butcher this name, Ignaz Semmelweis found that childbirth fever was spread by physicians who didn't wash their hands. So until, say, the 17th century, um, physicians weren't washing their hands when they were delivering babies, which could make, of course, the mother sick because that's a giant gaping wound. Um, so he realized that surgeons should maybe wash their hands. Louis Pasteur, inspired by Bassi, found that a protozoan called a disease in silkworms. So he's credited for saving the silk industry in Europe. And this is a name you definitely need to know. A man named Joseph Lister. He discovered that carbolic acid or phenol killed microorganisms, and so he would use these to treat surgical wounds. So in Victorian England, um, they used to have these amphitheaters and they would perform live surgeries. And it got to the point where if you were going to medical school, you were required to attend a couple of these, but the fatality rate of these live surgeries was very high, almost guaranteed you weren't going to survive it. Um, but we didn't know a lot about medicine. And so they would be performed really quickly. And Joseph Lister, during this time, um, realized that they needed to treat surgical wounds with something that killed microorganisms so people would survive. And then Robert Koch is one of the founders of modern bacteriology. He identified the causative agent for tuberculosis, cholera, and anthrax. And so he came up with Koch's postulates. And basically, this is how we learn about different microorganisms. So there's four different postulates. The microorganism has to be present in every case of the disease that you're studying. So he studied it in cows. Um, what he would do is he would take a cow that was sick, or he would infect the cow, and then there would be a healthy cow. So the microorganism would be taken from the host with the disease and grown in a pure culture, think like a petri dish. And then it would need to be reproduced when a pure culture of the microorganism is inoculated into a healthy host. So they would infect a healthy cow. Um, and then once that cow got sick, you have to recover that microorganism from the experimentally infected host. We'll talk about this more next week if you need more details. So there's another term in medical history. It's called the magic bullet. And you need to know that there is no magic bullet. Once scientists found that disinfectants killed microorganisms, they wanted to find a chemical that could kill all disease pathogens in the body, the magic bullet, but without harming the human host. So this is the foundation of modern pharmaceutical treatments and medicine, but unfortunately there's no medicine, there's no chemical that can kill disease, like every disease in a living human, because strong medicines have such harmful side effects. It would end up being um, detrimental to humans. All right, now we get into the meat and bones. How are microorganisms named? So we are going to play word association and learn prefixes, suffixes, and root words so that you can look at a microorganism and break it down and understand it. So the prefix are the letters added to the beginning of a word, like micro, meaning extremely small. The root word is the main word, like bio, which means life. And a suffix are the letters added to the end of a word, like ology, which means field of study. So if you look at the word microbiology, it would be the study of extremely small living organisms. There's certain suffixes and prefixes that you're going to want to know as we begin microbiology. And we'll go through these. This is just a couple examples. Microorganisms have a first name and a last name, like humans. The first name is the genus, which is capitalized, and the last name is the species, which is all lowercase. Um, Once you begin dipping into these studies, you'll recognize if you see um, coccus, it means shaped like a spear or a ball. That's a suffix. Or you may see the prefix staphel, which means clustered together like grapes. And aureus is a term that means a golden yellow color. So if you were to see the microorganism Staphylococcus aureus, it means that if you stick that pathogen under a microscope, it's a golden cluster of spheres. And that's how we can identify Staphylococcus aureus, which causes staph infections, skin and wound infections, and toxic shock syndrome. So bear with me. We'll come back to that. Let me touch on this. So whenever we studied anatomy, we studied mostly um, eukaryotic cells. Eukaryotic, like us. These are more developed. These are human cells, animal cells, protozoa, and algae. Um, Eukaryote means true nucleus. And most eukaryotic cells have a nucleus or several nuclei and membrane bound organelles that carry specific functions. Red blood cells are the exception in that they don't have a nucleus once they enter the bloodstream. And eukaryotic cells are 10 times the size of prokaryotic cells, they're much more complex. Um, Whenever we talk about kingdom Monera, prokaryotic cells, mostly bacterial cells, and cyanobacteria, which is blue-green algae. Prokaryotic cells are much smaller and less complex than eukaryotic cells. This is an example of that. The cell on the left is a bacterial cell. Prokaryotic cell, it's got a long flagella to help it in movement, and the image on the right is the eukaryotic cell like we have. There's 5 kingdoms of life. You'll want to know these. Kingdom Monera, or Prokaryote, which would be bacteria. Kingdom Protista for protozoa. Kingdom fungi for yeast, molds, and fungi, kingdom plantae for plants, and kingdom animalia for animals and humans. We have the order of classification of a species. This goes kingdom, division or phylum, class, order, family, genus, species. And this is how every living thing is categorized in science. There's a way to remember this. There's a mnemonic called King Philip, so kingdom phylum. King Philip came over for green soup, if that's a way you want to remember that. Or King David, either one. I'm briefly going through this. It's very important that you read your chapters. Within microbiology, there's different divisions. And the disclaimer here is that not all microorganisms cause disease. Many are very beneficial to our daily life. We have microorganisms that live on the surface of our skin, in our gut. Um, so when we're talking about microbiology for funeral service professionals, we're taking a look at disease causing microorganisms. So it's going to be less broad. The divisions that we're going to study is bacteriology, rickettsiology, virology, protozoology, and mycology. So bacteriology, the study of bacteria. Bacteria are prokaryotic, one-celled organisms from kingdom Monera. They are free-living organisms or parasites, and they multiply by binary fission, which basically means think of a sphere and just like pinching it with your fingers until it becomes two spheres. I'll show you all this when we meet again. So very important to know because for the NBE, for MATS, for DAD, you want to know that binary fission, that type of reproduction, is significant to bacteria. Um, Pathogenic bacteria cause the majority of infectious diseases, and bacteria are 10 times larger than a virus. And then we have mycoplasmas, the smallest and simplest self-replicating bacterium. If you look at them under a microscope, a mycoplasma colony is going to look like a fried egg, or I guess an eyeball if you look at it. There's no cell wall. And here's a test question. Mycoplasmas are the smallest free living organism. So we will talk about viruses, which are the smallest organism, but mycoplasmas are the smallest free living organism. Colonies of mycoplasmas form a fried egg shape, and mycoplasmas are responsible for UTIs, uh, primary atypical pneumonia, which is walking pneumonia, stillbirths, infertility, spontaneous abortion, and AIDS-related infections. Ricketsias are rod-shaped. So we'll talk about that in chapter 22, the physiology and morphology of bacteria. Um, but they can be spherical. They can change shapes. They're gram-negative, which refers to the way that they show up on a slide under a microscope. They'll stain differently. Um, Ricketsias are intracellular parasites. So they can only reproduce within a host cell. They're usually transmitted through arthropod vectors, which are going to be, like you see over here, ticks, mites, lice, and fleas. They are intracellular parasites within the arthropod, which is the living vector that bites and has to transmit it that way. Um, so it's also a biological vector. Um, Rickettsias cannot survive outside of the host cell, and they're responsible for Lyme disease and Rocky Mountain spotted fever. Those are the main ones that we talk about. Chlamydias, not the STD chlamydia. Chlamydias are non-modal, gram-negative, intracellular parasites that manifest like viruses, but they're classified as bacteria. They can replicate in the cytoplasm of host cells. They cause eye infections, genitourinary infections that are spread through mucous membranes. Um, Birds are going to carry a lot of chlamydias, like avian chlamydiosis or chlamydia psittaci, which is parrot fever. So when we say chlamydia, we're talking about more than the STD that we think of when we hear that. For kingdom Protista, we'll study protozoa, which are one-celled eukaryotes. Eukaryotes meaning that complex cell like ours. Most protozoa do not cause disease, but certain ones cause very severe diseases like toxoplasmosis, malaria, amoebic dysentery, brain infections, and infections in the skin and lungs. There's 4 classifications of protozoa: flagellates, amoeba, sporozoans, which means they produce spores, and ciliates. And for fungi or mycology, this will include fungi, filamentous unicellular and multicellular organs lacking chlorophyll, usually bearing spores. They often grow on decaying or decomposing matter, which means they are saprophytic. And this will include yeasts and molds. Viruses are infectious parasites. They're the smallest disease causing microorganism. They can infect almost any living cell. A virus is a piece of genetic material wrapped in a protein, and it contains one type of nucleic acid, either DNA or RNA. And then we have viroids, which are similar to viruses. They only contain RNA. They don't have a protein coat. And most diseases caused by viroids are going to occur in plants. But there's one deadly viroid specific to us, known as hepatitis D, as in dog, which it can be enclosed in the capsule of the hepatitis B, as in boy virus, and it can be transferred to the embalmer through blood pathogens. Prions do not contain RNA. They don't cause an immune system response. Um, they consist entirely of proteins produced by human genes, which means they can only multiply inside living cells. Prions are very terrifying because they cause spongiform encephalopathies, which means that they create holes in the brain. If you look at a brain scan, it looks like a sponge. They create huge holes in the brain. Um, Prions they're suspected to cause Crutchfield-Jakob disease, Alzheimer's, Down syndrome. They're suspected to also cause fatal familial insomnia, which is fatal insomnia. It's very deadly um, genetic disease. Prions will cause amyloid degeneration, and it can be inherited or transmitted through blood-to-blood contact. Examples being scrapie and mad cow disease. If you get an any embalmer group, they're always going to bring up CJD, Crutchfield-Jakob disease. The thing about this disease is it's very rare, and there's no way to diagnose it unless there has been a cranial autopsy. There could be suspected cases, and the hospital staff would be responsible to tell you it's a suspected case, but oftentimes it presents as um, dementia, and so they're not really gonna know. They're not really gonna know that. Fun fact: I believe um, a recent. It happened recently, within the past few years. The USDA is no longer required to test for prions in beef and meat. So take that as you will. Here's your chapter review. I'm going to go into chapter 22, anatomy of bacteria. These are old slides. I need to update them. I mean, they're still accurate. So how do bacteria multiply? Here's a good graphic of that. Bacteria multiply through a binary fission, which is a simple transverse fission, a method of asexual reproduction. um, Means division in half. It literally, see the parent cell on the left, the DNA duplicates, it divides into two daughter cells. Um, this will occur simultaneously, it happens at the same time. Um, a visible group of bacteria that grows on a solid medium is known as a bacterial colony. And if perfect conditions are met, which is not often in a controlled lab, some bacteria can reproduce every 20 minutes. Let's talk about the morphology of bacteria. There are three basic shapes. Those shaped like a sphere, which is a coccus. You can remember it because it starts with a C. You can think circle. Those shaped like a rod, bacillus, and those with a spiral shape, spirilla. So those are the basic shapes. There's several more shapes, but these are the ones that we're going to talk about. And there are three basic arrangements: diplo, which would be a pair; strepto, which would be a chain of bacteria; and staphylo, which is a cluster. So um, this will make more sense as we take a deeper dive into the study of bacteria, because think of strep throat, which would be Streptococcus pyogenes, and we'll learn that. Um, Sorry, I just got a text from my mom. Once you become familiar with these prefixes and root words and suffixes, it'll make a lot more sense. So let's talk about bacteria with a spherical shape. This will include a coccus in their name, or the plural cocci. Spheres are arranged in many different ways. Diplococci would be, I'm sorry, let me pause this for just a second. I need to look at something. apologize. I'm not just randomly texting. Y'all know my mom is going through restaging and they found something on a scan, so she's going to have to stay another night in Arkansas. Okay, anyways, I was oversharing. So let's come back to bacteria with a spherical shape. Include the word coccus in their name, the plural cocci. Spheres arranged in pairs are diplococci. Spheres arranged in a chain are going to be streptococci. And spheres arranged in a cluster are staphylococci. These are going to cause diseases like strep throat, food poisoning, scarlet fever, wound infections, think staph infections, think MRSA, and toxic shock syndrome. Bacteria shaped like a rod will include the word bacillus in their name, plural bacilli, and rods paired together are diplobacilli. If they're in a chain, they're called streptobacilli, and these cause diseases like typhoid, diphtheria, and lockjaw, which we know as tetanus. Bacteria that are spiral and helical shaped are named spirilla, shaped like um, oh, sorry, I saw that bold. So this could be a board question. It was on board prep. Spiral and helical shaped bacteria. They're named spirilla, and you could see them described as corkscrews. So think. I'm sure most of y'all are familiar with like a cork screw, like you open up a bottle of wine with. That's the shape that we're talking about. It's on the image here. Spirochetes are going to control their movements by turning like a corkscrew. And there are also curved rods that are known as vibrios. These will cause diseases such as Treponema pallidum, spirochaete, or syphilis, and Borrelia burgdorferi, which is a Lyme disease. All right, so almost all prokaryotic cells will have cell walls. Their primary purpose is to prevent bacteria from rupturing when the osmotic pressure inside the cell is different from the pressure outside of the cell. The human body will defend itself from bacteria by producing antibodies that will attack the cell wall of a bacteria, but not all bacteria will have cell walls. For example, mycoplasmas, which are the smallest known bacteria, they do not contain cell walls. So let's go back to the basics. A cell membrane or a plasma membrane will line the inside of the cell wall in many bacteria. The cell membrane is composed of phospholipids, which are units of fat and clusters of phosphorus. So this means that um, soaps, alcohols, and quaternary compounds can kill bacteria if they break down the phospholipids in that cell membrane. If the cell membrane is damaged, the inside contents of the cell are exposed and the bacterial cell dies. Um, in eukaryotic cells, like the ones we have, cytoplasm holds the organelles in the cell. In prokaryotic cells, there are not um, organelles, but they do contain cytoplasm. And it resembles the cloudy elastic fluid. It's rich in enzymes, carbohydrates, and lipids. Prokaryotic cells secrete a sticky gelatinous coating known as glycocalyx, which will surround the cell wall. Um, this often will form a capsule, but only if it's organized and attached externally. If it's not, it's called a slime layer. And capsules, if they're able to form, allow some species of bacteria to survive in adverse circumstances because it gives them an extra layer of protection. Many bacterial cells are flagellated, meaning that they have flagella, which are those long whip-like structures that help propel them and allow them to move. The only human cell that has a flagellum is the human sperm cell. Some bacterial cells are covered with structures known as fimbriae and pili, which are much shorter than flagella, but they allow them to attach and adhere to surfaces. Some bacteria can form endospores, and the ones that do are very dangerous because if the bacteria can create an endospore, it basically forms a resistant form of itself that can survive for thousands of years. Sporulation is the process of forming an endosphere. They're usually found in gram-positive bacteria, and endospores are generally harmless until they germinate into that vegetative state. That could be a board question. I know it's a maths question. Um, Diseases that can cause endospores include, or bacteria, sorry. Um, Bacteria that form endospores can cause anthrax, tetanus, gangrene, and botulism. Clostridium is often in the names of these, like Clostridium tetani. I'm trying to think. My brain's not all here, but we'll go through these as we see them. Oh, yes. Clostridium tetani, Clostridium perfringens, which can cause gangrene. So this is significant to us as embalmers because endospores are extremely difficult to kill, if even possible. They're very resistant to disinfectants. So what's embalming? Disinfection, preservation, restoration. And because human remains are only disinfected, not sterile, endospores can still remain in the body and they can still be present in human remains even after embalming. You can also transmit um, certain pathogens if they form endospores through your embalming instruments, meaning if one of your bodies has tissue gas, which is Clostridium perfringens, um, true tissue gas, um, if you've ever smelled tissue gas, you'll never forget that smell. Um, but you can then give other bodies that come into your prep room tissue gas because that can remain on your instruments. So you do want to make sure you do your best to sterilize your instruments. Um, and we can sterilize inanimate objects like our instruments, but we can't sterilize a body. How do microbiologists study microorganisms? Through fluid suspensions, smearing, and staining. So fluid suspension, think like a microscope slide is typically what we first think of. So it's suspended in a fluid. There's two different methods. There's the hanging drop technique, which is in this image on top up here. There's going to be like a dip in the slide, and so you can drop a drop of liquid with the specimen, and then you seal it with a cover slip and petroleum jelly around it. That's the hanging drop method, or the wet mount technique, which would be a ring of petroleum jelly on the cover slip, which goes on top of the specimen. You drop the specimen onto it, and then you place the cover slip, which has petroleum jelly around the edges. That's a wet mount technique. There are also smearing, um, which is a sample of blood or a specimen spread on a slide or on the surface of a cultural medium. This is like um, if they're taking blood samples and they smear it onto a card to send off to a lab, or if they collect stool samples, which is where you smear stool onto a card and they send it to a lab. Bacterial smears have to be made and fixed. So they're made by evenly spreading bacteria on a clean slide and a drop of liquid, which is sterile water typically. And then we have what's known as staining. And so staining, this concept, is how we um, identify and group bacteria. A common dye that's used to observe DNA or RNA is called methylene blue. And so dyes with a positive ion charge are methylene blue and crystal violet. And so basically, if you drop methylene blue onto the sample, they're going to stain any alkaline structures or basic structures in the cells. And those are dyes with positive ion charges. Dyes with negative ion charges or acidic charges um, are acid fuchsin, Congo red, and cosin. You actually would need to know if you see like Congo red or something that's a dye. These are used to stain positively charged cellular proteins. Sudan black will stain fats and cells. And so these stains are used so that the entire cell and its structures can be seen clearly. And it's one of the most important methods of classifying bacteria into gram positive and gram negative categories, because based on whichever way they stain, they have certain characteristics. This is the process, which I'm going to um, kind of skip over. We can come back to this next Wednesday during current issues, but you're not really going to be asked this because you're not going to be in a lab preparing this. It is good to understand the general concept. So we'll return to that. What you need to know are these things. How do bacteria multiply? Binary fission. What are the three basic shapes? Um, Kochi, Bacillus, Spirilla. What are the three basic arrangements? Diplo, Strepto, Staphylo. Describe the significance of encapsulated bacteria. Um, Compare and contrast cell wall and cell membrane, and motility of bacteria. So we'll return to that because I'm trying to get through this. Let's see how long this one is. 10 slides. I can get through it. So this is physiology of bacteria. So we just talked about anatomy or morphology. So the shape of, this is the functions of. There are certain requirements for microorganisms to survive. They have to have certain ways to obtain nutrients and food, whether they can survive in oxygen or in the absence of oxygen. Moisture requirements. Most microorganisms require moisture. That's why they survive so well in the human body. There's temperature requirements and pH requirements. All microorganisms have some source of nutrients or food source in order to survive. Um, there's two different types: autotrophic bacteria that are capable of self-nourishment, so they make their own food, or heterotrophic, which requires complex organic food from a carbon source to grow and develop. Um, Heterotrophic bacteria, they do cause disease, but they're also good bacteria too. They're integral to fermentation, to producing wine, bread, cheese, beer, and sauerkraut. Um, and then autotrophic bacteria they can be good too because they produce 20% of the world's oxygen. There's three categories of heterotrophic bacteria. You do need to know this: strict or obligate. You can use those terms interchangeably. If they're saprophytes, and they're strict saprophytes, they can only survive on dead or decaying matter. Strict or obligate parasites are completely dependent on a living host. If they're facultative, they can adapt. If they're obligate aerobes, they can only live in the presence of oxygen. If they're obligate anaerobes, they can only live in an environment devoid of oxygen. Microaerophilic organisms require very little oxygen. If they're facultative, they can adjust to changes in oxygen levels, and aerotolerant organisms can grow in the presence or absence of oxygen. Generally, disease-causing bacteria require a lot of water, so this is why they flourish in the body's tissues. Uh, Molds and yeasts differ because they require less moisture and occur on the surface of the body. So think fungal diseases like athlete's foot. All right, some bacteria require pH requirements. So remember the pH scale: um, 0 to 14, 0 to 6.9 is acidic, 7 is neutral, 7.1 to 14 is alkaline or basic. The human body is slightly alkaline with a pH of 7.4. And so bacteria that cause disease in humans thrive at a neutral pH. However, there are bacteria like Helicobacter pylori, which cause stomach ulcers. They survive in the acids produced in the stomach. And there's also yeast and molds that grow well in acidic environments like the vagina. Um, the vagina has a very acidic environment, so that's why they're more prone, or that's why the female body is more prone to yeast infections. Yeast and molds don't contain chlorophyll, so they prefer dark areas. There are some types of bacteria, like cyanobacteria, that will utilize sunlight as a food source, so they thrive in light. Um, you'll notice when you go back into the classroom next week, if you look up, we have some UV lights, which were installed during COVID, the COVID pandemic, and 2020 is when they were installed, my first year here. Um, and that's so at a certain time at night, they cut on because UV light can kill certain microorganisms. And then we have osmosis and osmotic pressure that helps us control microorganisms. We'll talk about this next week. Here are some more definitions that you'll need to know. Microorganisms can live in the presence of each other, and there's different ways that they um, associate. So these are definitions. There's antagonism, which means they're in mutual opposition of each other. Commensalism, which is where a bacteria will benefit from a host, but the host is not harmed. Mutualism means there's mutual benefits. So we have bacteria in our gut biome, and they get to live happy in our gut, and we benefit off of that, because if you have a diverse biome in your gut, you actually are in much, much better health. That's why when you take antibiotics, you feel so sick, because it's stripping even the good bacteria. Um, Parasitism is when a parasite benefits and the host is harmed. Symbiosis is when two different species live in close association. So they're just living beside each other. No one gets hurt, no one benefits. And synergism means that when two organisms come together, they have a benefit that couldn't be obtained otherwise. So I kind of flew through it, but it is just an introduction. And I'll be happy to answer any questions, and just make sure you're in your readings. We can review this next week if you have any questions. Why don't I stop sharing? I hit that, I end the call. Okay, sorry. Here we go. Okay. If you have any questions, please feel free to get in touch with me. Um, Next week, we're going to be in chapters 23 and 24 of Micro, um, which is control of microorganisms and the cycle of disease. Um, in embalming next week, we're going to do chapters 1 and 2, an intro, and then some more terminology. So I will try to scan in at least chapter 1 for y'all for next week. And then, of course, next week we will finish up sociology. All right. I will let y'all go. Um, Let me know if you have any questions. Thanks for attending today.
Have a good one.
Thanks.
See you later.
Bye.